All right, so Cameron mentioned he was the last man between you and beer. I am the last woman between you and beer. I'm Elizabeth Phelps. I'm just newly joined the Department of Psychology here at Harvard University. My research is exclusively on humans, so I kind of took the translation part of this seminar quite seriously. And so I wanted to start with just a few points, and then I have some questions that I've written down for the speakers, and hopefully we'll have just a couple minutes for questions from others. But when I think about how we do translational research, right? There's a ton of good reasons to study animal models in terms of trying to understand the neurobiology and develop new drug treatments and things like that. But the question, of course, is how do you do the translation correctly? And this, of course, is the $64,000. Well, that's not very much these days, really. Like what was that in the 50s? That's, of course, the million-dollar question. How is it we know that we're actually doing translation well? And there's a couple ways we can go about this. So, in my lab, we try to sort of start with looking for behaviors that are as similar as possible, using behavioral paradigms that are as similar as possible to what has been done in the animal model. And so we've done a lot of work with Joe Ledoux. I've collaborated with Joe Ledoux for almost a quarter century at this point, and sort of very similar threat conditioning, Pavlovian threat conditioning procedures, trying to look at behavioral outputs that are essentially the same, essentially physiological markers of defensive survival circuits, things like autonomic nervous system responses, which you can actually do both in animals and humans. And this is a really obvious way to do this. This, of course, leads to the problem of, what does this have to do with what we're doing outside the laboratory? So one thing we're missing, of course, is subjective reports. And when we talk about psychiatric disorders, we are primarily relying on subjective reports, sometimes reports of others or actual behaviors that they engage in, but often subjective reports about the nature of the disorder. And Joe in particular has talked a lot these days about how it is we can think about the ways people say they feel and how does it link to these sort of biological markers of things like threat reactions. And the links are sometimes good, but they're often not very good. So I do think one thing we're always missing with the animal models is linking to what we think is one of the most important variables in studying psychiatric disorder, which is how do people say they feel. Another thing that's been used in sort of translation from animal to humans, and this was talked about today by the speakers, is this notion of behavioral phenotypes of psychiatric disorders. And so here we try to say, well, here's a few characteristic behaviors of this disorder, and now let's just look at animals and see if we can create those behavioral phenotypes in the animals, doing things like genetic manipulations, or just looking at individual variability, and then assume that has something to do with the psychiatric disorder we're measuring. So a couple of those we talked about with exploration, for instance, in rodents. Bob talked about this. What's interesting, I think, is sometimes these phenotypes, for autism, the sort of lack of social engagement and things like that seems somewhat obvious, but sometimes these phenotypes have not been kind of, when they're actually behavioral markers you can measure aside from feelings, have not been investigated across species as much. So exploration, for instance, this is something that seems very obvious that we could use some of the tracing technologies that we've heard about today to see if there's really a big difference in anxiety disorders as exploration as a measure. In my laboratory, we looked a little bit using laboratory paradigms of exploration, sort of decision-making paradigms, and haven't actually found they've been linked to things like trait anxiety. So we have to kind of also be able to make those clear links. So one question I want to talk about is how do we bridge this divide with subjective reports? And are behavioral phenotypes, physiological measures, are they good proxies for this or not? And when do we know whether that's the case? When we translate from animals to humans, we're also thinking about translating the neural systems from animals to humans. And so one thing we can do is do similar techniques across species. This, of course, we're very limited in. You can do functional imaging in other species, and you can do it in humans. You can do lesion studies in humans. They have limitations. You can do them in rodents. You can do pharmacological manipulations in humans and other species. But again, you have to figure out which behavior you're looking at. And then, of course, EEG, and with things like intracranial EEG, of course, you have limitations in humans as well. But obviously, the reason you have animal models is because you have different scales and interventions that you can do in animal models that you can't do in humans. The one advantage to humans these days is that you can actually then, you're often looking at more whole brain networks, not that you couldn't do this in animal models, but because of something like functional imaging, we're actually getting whole brain networks very often, and that's something we talk about a lot more when we look at human brain disorders than other species. So this again leads to this question: when we have this much greater level of specificity in animal models, how do we bridge the divide to know that that specificity is going to be applicable to the disorder that we're interested in? And I think these are the challenges you have to think about when we translate from animals to humans. And so now I want to invite the speakers up. I have specific questions for each of them, and then I'll open it up to the audience. So if anyone wants to come up.